Hello, everyone. Must be having good time with your families. I pray for all of your health and best of time with your family. As you all know that we are now gradually moving towards the terminal topics of the medical microbiology. And today we will discuss another group of microorganisms which do not occur in the form of cocci or in the form of rods or they do not make spores. They have a different morphology. So before moving on to the actual topic, we'll deal with the group of microorganisms which, sorry, which belongs to what we call as the spirochetes. They are the spiral-shaped microorganism, and their makeup is different with compared to that of the gram-positive bacteria or gram-negative microorganism. Before we move on to the specific genus or the species of this group of microorganism, what we call as the spirochetes, let's comprehend or try to understand the structure of this organism. This is the diagrammatic representation of this spirochetal cell. And as the name designate or indicate, you can really imagine that this is an organism which occur in spiral shape. That's why they're called the spirochetes. They make coils and they occur in spiral form. So this is what we call as the spirochetes. So if you look at the structure of the spirochetes, they have what you call as a specialized structure, which we have not seen before in any of the gram-positive or gram-negative cell. And this is what we call as the axial filament. Axial filament are what we call is the endoflagella. Because the flagella, which is you see in this case, or the axial filament in this case is of pink in color, it runs across the entire body of the spirochetes and it is enclosed in the sheath. So that's what we call as the endoflagella, because the flagellum of the spirochete is enclosed in a sheath and it runs across the spirals of the body of the spirochetes. So this is what we call as the endoflagella. So if you look at this, this is the cross section of the body of the spirochetes. And if you look at this, that we have this what we call as the axial filaments. This entire is the axial filaments. And we have the what we call as the structure inside what we call as the endoflagellum. Because the endoflagellum of spirochetes is enclosed in the sheath, we call it as endoflagella because of this structure or the covering of this microorganism, the flagella of the microorganism. Now, the other structures that we can see in the spirochetal cell, it has this cell membrane. If you look at this, it has this cell outer membrane. And in between the outer membrane and the cell membrane, we have a broad space, as we have in case of gram-negative cells, what we call as the periplasmic space. So technically speaking, the spirochetes, they are gram-negative cells, but we need different strategies because they're too small. We cannot see them with the help of a microscope. We have to adopt the other strategies in order to visualize this microorganism under the microscope. So they have the well-developed periplasmic space. They have the flagellum, which is enclosed in the sheath, what we call as the endoflagella. And if you look at this, you see this outer membrane, it contains many antigenic proteins, like the proteins of the gram-negative cells. So well-developed periplasmic space, endoflagella, and the outer membrane, they are diverse in the antigenic proteins. So they have many proteins. And that form the basis of the many serovars that we can see in case of some of the species of the spirochetes. So the objective of this slide is just to make you understand that the spirochetes, as the name designate, they are the spiral-shaped organism. They cannot be compared with that of the cocci or the rods, but they are in fact, they are gram-negative. And as you have seen that it has a well-developed periplasmic space, it has a flagellum which is enclosed in the sheath, what we call as the endoflagella. So the flagella of spirochetes, that helps. You see, the host organism, it is motile through the undulation, or what we call as the undulation of the axial filaments, which is under the sheath. So the organism, they are motile with the help of the undulation of these axial filaments. They have the periplasmic space, and they are small. We cannot see them. They are thin, small. They make coils. So we cannot see under the microscope, under the ordinary microscope, with the help of the Gram stain procedure or the SLFA stain. We have to go for some of the other stains that we will discuss. So I'm sure that you now comprehend the spirochetal structure. Now, if you look at the properties of these spirochetes, as you have seen, they are the thin-walled, flexible spiral rods, and that makes sense because you see there, the spiral can only be made with the help of the thin wall. If they are rigid and thick wall, if the organism is rigid cell wall, it has a thick cell wall, it cannot be in the form of coils. So the flexibility is because of the thin wall of the cell, and they are motile with the help of axial filaments, what we call as the endoflagella. Typically, as they have the well-developed periplasmic space, they are gram-negative cells, and majority of these organisms, the spirochetes, they are the free-living, they are the commensals. So thin-walled, flexible, gram-negative cell wall having endoflagella and the well-developed periplasmic space. These are the major characteristics of the species of the spirochetes. Now, we have three clinically significant genera of the spirochetes. Number one is the Treponema, and then we have the Borrelia, and then we have the Leptospira. Three main spirochetal genera are Treponema, Borrelia, and Leptospira. We'll see each of these genera first, and then we'll move on to the specific species. Now, if you look at this slide, again, you see the artistic representation of the architecture of a spiral cell. This, the endoflagella, we have the periplasmic space, endoflagella, this is the what we call as the cell body, and we have the outer membrane. Now, this is the staining, what we call as the silver staining. This staining is a specialized procedure that is used to see the microorganism because they're very thin. We cannot see in the microscope. So we'll have to adopt other strategies that can enhance its visualization of the microscope. So this Treponeme, all these three genera of spirochetes, the Treponema, the Borrelia, and Leptospira, they are differentiated, or they are designated, or they are identified based on the coils that they do have. Either they are tightly coiled, they are loosely coiled, or they have some hooks at the end of the spirochetal cells. So based on to the configuration of coils, loose or tight or intermediate, or they have hook on sides or not, we identify these microorganisms. The Treponema, typically speaking, they have evenly spaced. They are evenly spaced, thin, and they have just, the tight coils. And the species of significance of the Treponeme is the Treponema pallidum that causes syphilis. We will take the species in detail. Now, the Treponema pallidum, it spread, its transmission is through the intimate contact. The sexual transmission is there, and it can also cross the placenta. So it can cross the placenta, and it can be spread through the help of sexual contacts. Now, the organism is so small that we cannot see under the iris microscope with the Gram stains procedure. So we'll have to go for what we call as the dark field microscopy. Or we do another staining characteristic, what we call the silver impregnation. Or we can use the immunofluorescence stains to see the microorganism under the microscope. Now, the Borrelia, they're a little larger, and they accept the GENERA stain and the other blood staining procedures, and we can see them under the standard light microscope. But the Treponema and the Leptospira, they are so small and they cannot be seen. We have to go for the dark field microscopy or silver impregnation, or this is what we call the silver staining. This staining procedure is it's very sensitive staining technique, And this technique is mostly used in case of visualization of protein fragmentation under the UV light. Whenever you go for the Western blot, that is transfer of the protein onto this nitrocellulose membrane, and we have to see under the UV lights, so we can go for the silver impregnation. It's very sensitive staining technique. So taponims, they are evenly coiled, tightly coiled cells. They cause a disease known as syphilis, which is sexually transmissible disease. It can also cross the placental barrier. And because of the small size of the taponym, we cannot understand, visualize the microorganism under the ordinary microscope. So we'll have to go for the dark field microscopy or adopt another staining procedures, the silver impregnation of the cell, or the immunofluorescence staining procedure. Now, the Borrelia, if you look at this, although you see the number of coils, it may varies, but generally we can say that it has 3 to 10 coils. They are large and they're loose coils. Look at it, they're loose, they're not that tightly coiled compared with that of the taponimal cells. We have 2 species which are clinically significant in the Borrelia. The Borrelia burgdorferi, which causes Lyme disease, and the Borrelia recurrentis, which causes relapsing fever. So 2 organisms, Borrelia burgdorferi, that causes Lyme disease, and Borrelia recurrentis, which causes relapsing fever. Now the transmission of these Borrelia, it is with the burgdorferi is transmitted through the tick bite, whereas the recurrentis is transmitted through the louse bites. So this is what we call as the arthropod-borne viruses, because they have a source which belongs to the arthropod category of the animal kingdom. Now this organism, it is comparatively larger in size. It is more visuable, so we can stain that with the help of the gene cell staining procedure, which are other blood staining procedures. All right. The next genera is the Leptospira. Now, if you look at this, you can really differentiate the configuration or coiling pattern of the Leptospira compared with that of the taponims and the Borrelia. Borrelia, they're larger. Taponims, they're evenly coiled, but they're thin. And this organism has numerous fine, regular, tight coils or spirals. If you look at this, numerous, very large, tightly coiled, very close coiling, not that much of that. Sometimes you can see the one end or both end of these Borrelia, they are curved. So Leptospira, the end of Leptospira can be curved, one end or both of the ends. We'll see that in another slide. So the Leptospira, they are tightly coiled, their ends may be curved, and the important species in Leptospira is the Leptospira interrogans. And this causes a disease what we call as the leptospirosis. Now, this organism can be transmitted through food or drink which is contaminated by the urine of the infected animals, like rat, dogs, cows, pigs. And this organism, being very small in size, again visualized under the dark field microscopy. So this is how all these 3 clinically important genera will look like if you want to see them under one slide. This is the diagrammatic representation of the spirochetal cell. This is the cell of the taponym. This is Leptospira. This is Borrelia. So now you can see that the Leptospira, they are tightly coiled. Taponims, they are loosely coiled. And the Borrelia, they are more loosely coiled. So the tightly coiled, loosely coiled, and we have the intermediate configuration of these cells. They have the periplasmic space, they have the endoflagella, cannot stain with the gram stain procedure. So they are typically gram negative, but we cannot see them under the microscope because of the small size. We will today take one of the very important organism belong to the genera, what we call as the, we'll let you know that later on. But you see, this organism of this genera of treponym It is, you see, transmitted through the urine that contaminate the water or the food material. And contaminated urine comes from the animals like rats, the cats, the dogs, the cows. And if the organism is tightly coiled, it has some hook at one end maybe. It is excreted in the urine of the rat or cat and dog. And if you are really come in contact with that contaminated water or food material or soil even, and you have some abrasions in the foot and you are working or walking barefooted, the organism can enter into your body. So the source is the arthropod, the material is the urine, and even you see the rainwater, marshy area, this area, even you see the dog, it can excrete this organism in the urine, and this can contaminate the water. And if you are walking barefooted in the water and you come across some injury and the organism or chances of organism for entrance in your body. So this is what we call as the genus of the member of Leptospira, and it causes a disease what we call as the leptospirosis. It causes a disease what we call as the leptospirosis. So what is the leptospirosis, frankly speaking? You see, we have the contaminated urine, we have the contaminated urine, and if you examine the microscope, it's the bacteria what we call as the Leptospira, tightly coiled, may have a hook at one end, if you look at this. And then you see it entered through the cut skin in the body, and it is a pathological bacterial infection, which is basically transmitted by the animals to the humans. The source of infections are the wild or the domestic animals, or even the okay. Now, but the WHO said the definitions of leptospirosis is almost the same. I will not go into the detail, just to make you understand. It's a bacterial disease, of course, and it can affect both the animals and the plants, the animals and the humans. And the other stage of the disease, we will not can really differentiate because it has just the generalized signs and symptoms like fever, headache, muscles, pain, chills. Redness of eye is very characteristic with this. This again is common, and there is jaundice. This again is very characteristic, and hemorrhages on the skin and the mucous membrane. It can go even to the lungs, bleeding, vomiting, diarrhea, and rashes. So initially generalized signs and symptoms. There must be history of, you see, the individual having exposed to the contaminated waters. Some of these, see these swimmers, the swimming pool sometimes is contaminated with the urine, and swimmers, they can really have this infection. The cases has been reported, even epidemics in this case. Now the Leptospira itself, the important species of which is the Leptospira interrogans. This is tightly coiled microorganism, and it is really curved at one end. If you look at this, it is curved at one end. Look at this. This organism is curved at one end and both ends. Now, curved at one end is the Leptospira interrogans, and we have another species, what we call it the Leptospira biflexa. This is has a hook at both ends. So we have one hand, we can have both ends. One end is interrogans, tightly coiled. Both ends are the biflexa. The interrogans, because of the variation in the antigenic proteins of the cell membrane, it has 20 serotypes, 200 serotypes. Now, 200 serotypes, they are really prevalent in specific species and specific areas. So based on the antigenic differentiations in the cell membrane of the Leptospira, we have more than 200 serotypes of the Leptospira interrogans. Now, the immunity to Leptospira is serospecific. One strain, serospecific, cannot provide protection against the other strains. So it is serospecific. Now, both species, the interrogans and biflexa, they are serologically, genetically, and physiologically distinct. So one would not provide protection against the other ones. And even within the interrogants, we have 200 serotypes. So the one serotype's infection, if immune, cannot provide protection against the other serotypes. All right. If you look at the epidemiology and the transmission of the disease is almost, it's a zoonotic disease, and its transmission is associated with the, or it is present in the wild animals like rats, rodents, skunks, and foxes, and it is also present in the domesticated animals. So the wild and domesticated animals like horses, dogs, cattle, pigs, and dogs are important reservoirs, particularly in the Western society, where they used to keep dog and puppies inside the premises, living premises. There are big chances that they may be having infection and they can contaminate the environment. So both the wild animals and the domesticated animals, they are the source of this infection. They can also have this infection. And the disease leptospirosis, it's mainly concentrated in the topical area, particularly during the regional season. This is not that prevalent now in the United States. It used to be. But now you see the area like where the heavy rains like Vietnam, Philippines, Thailand, they are more exposed. Even the marshy and the highly rainy area in our society also. Excuse me for a while. Now this is the diagram just to, picture just to illustrate the leptospiral transmission. The bacteria, which is Leptospira, infected animal, it excretes the Leptospira in the urine. And this urine may contaminate the water, soil, or food, or this urine may expose to the urine or contaminate the water, soil, and you are exposed. Once you are exposed to the, actually the sources, the rodents are large animals, contaminated environment like water, like contaminated food material, and you have some injury. If you are even you see some something, you are swimming in the swimming pool and the swimming pool water has been contaminated with the urine of a leptospiral organism, you can get infection. All right. Now the the organism, if you look at the organism's mode of action, or whatever, say it's, the organism is shed in the urine of the infected animals. We are not sure. The Leptospira is excreted in the urine of the infected animals, and this organism can survive for months in the natural inclined soil. So the pH of the soil or water or food material does not have an influence on the organism's survivability. It can survive. It has all the potential to survive in both pH values. The immune infections, as we know, that it occurs whenever there is abrasions or some damage or some breakage in the skin and or in the mucous membranes, and it is contaminated with the animal urine or the environmental source containing the urine. So if your skin is intact, you are fully protected. But if your skin has some kind of breach because of some trauma, because of some mild or unknown abrasion in the skin, and you are exposed to the environment having Borrelia, the Leptospira cell, you can be infected. After entrance, the organism would circulate in the blood, and it will then, after entrance, it will multiply, circulate, and then it will lodge into the various organs and will cause the dysfunction. When it will go to the liver, it causes jaundice. It can lodge in the kidney, causes uremia. It can go in the lungs, where it causes the hemorrhages. It can go into the CNS, which causes aseptic meningitis. And in the eye infection, conjunctiva, the entire redness of the eye area. So the symptoms of the disease, they are specifically analog with that of the organ involvement. Liver involvement is there, we can have the jaundice, kidney uremia, lungs hemorrhages, CNS aseptic meningitis, eye conjunctival infections. The disease typically occurs, as you see, it's a biphasic disease. This means that the disease occurs in two phases. there is first initial phase or the early phase, what we call the leptospiremic phase. In this time, during this time, the organism is present in the blood. This is the early phase. Now, it appears in the blood and the CSF, and because of its presence, there is high fever, there's chills, intense headache, fever, and conjunctival suffusion, what we call as the redness that is scattered in all the conjunctival area. So this is the initial phase. In the initial or early phase of leptospirosis, there is leptospiremia, number one. Number two, the multiplication, release of certain virulence factors. It causes the fever, chills, headache, muscle pains, and organism through the blood goes to the conjunctiva, causes redness in the area. Now, this initial phase, it is resolved for some times, and the symptom that disappears and organism clears from the blood. Now, what happens? It then enters into what we call the secondary phase, or the immune phase. Now, in the immune phase, in the initial phase, the signs and symptoms were because of the presence of the circulating leptospiral cells who were multiplying, who were secreting the virulence factors. And they were lodging into the various organs like lungs, like kidneys, like liver, like CNS, like eyes. But in the second phase, it is the immune response that causes the signs and symptoms. Because the organism has been cleared, it is nowhere lodged in the body area. But the immune response is there. So sometimes later, the second phase of the disease starts, and this is what we call as the immune phase. Now, in the immune phase, the blood infection disappears, of course, due to the action of the phagocytic cells, the complement antibodies. And this is what we call as the aseptic meningitis, liver damage, impaired kidney functions. This is what we call as the Weil syndromes. You know, these syndromes, they are because of the immune response of the body. Now, what happens in the Weil syndrome? It's a cluster of symptoms, what we call as characterized by the kidney invasions, hepatic diseases, jaundice, anemia, and neurological disturbances. This do occurs in many of other immunological problems. So we have the initial phase, which is the leptospiremic phase, and we have the second phase, and the late phase, what we call as the immune phase, the biphasic disease. Now, this again to just make you clear more about the leptospiremic phase. There is a non-specific flu-like symptoms, headache, muscle pain, eye pain. There is chills followed by redness of the eyes. And this initial leptospiremic phase, it lasts for about 3 to 10 days, and there is the leptospira in the blood. That's why we call it the leptospiremia. Then in the later phase, what we call it the immune phase, fever is not responsive to the antibiotic in this area. And there is stiffness of the neck because of the involvement of the meninges, and inflammation of the nerves and eyes, brains, and leptospira in the urine. So in the acute phase, if you want to identify the organism, you will have to go for the blood specimen. If in the later stage, when there is immune phase, you will have to look the leptospira into the urine. And this is what we call as the Weil syndromes. It's a fulminant type, jaundice, pulmonary hemorrhage, acute abdominal myocarditis, and this is the immune response of the disease. Now, how about diagnose the disease? Of course, the history of the exposure of the individual and presenting signs and symptoms are almost clinical clues that can give you an idea about the disease. We can go for the dark field microscopy. We can go for the culture, but it needs some specialized ingredients in it, particularly the serum. And you can go for the serological test, serum antibody titers. Microscopic slide rotation test, we can see most often with the fast, specific, and effective test. We can do that. So generalized diagnostic procedures are there. Probably go for the culture of this microorganism. If we do, we can see we can make a smear, we can stain it with G-SUM stain or with the silver staining procedure, or we can see it under the dark field microscope. This is the early diagnostic procedures. We have the direct methods, we have the indirect methods. Direct methods, immunofluorescence staining, silver staining, which is, I would recommend that. This is really magic. Various methods of PCRs are there, but in the indirect methods, we have to go for the ELISA and the glutation test. So the diagnostic protocol is almost the same as we can go for the other bacterial species, depending on the acute phase or the chronic phase of the disease. The organism is present in the blood or urine, or it has gone into the immune stage. The treatment is penicillin G or tetracycline. And we can start with the fourth day of the illness, and afterwards it becomes less effective. And the live formalized Leptospira mixed with serum observed for the glutation and the lysis. You can take a slide. You can lay the Leptospira and we can mix with the serum. So this is commercially available. You will have to take it from the what you call it, the CDC and our other reference lab, and you can mix that and you can see the glutation test. Stain specific vaccine is stain specific, as we see. It is made from the killed cell. So one stain will not provide protection against the other stain. You will have to go for the stain specific vaccine. We can prevent after avoiding ourselves from the contaminated environment. This is the line of treatment. You see, if you really specifically proceed for the treatment of the Leptospira, there's mild leptospirosis, there's moderate to severe, and the antibiotic therapy. In mild cases, we go for the doxycycline, amoxicillin, azithromycin, and in moderate cases, we need the penicillin, ceftriaxone, or azithromycin. And this is for the 7 days and 3 days azithromycin. Right? This is a real good drug for this. You can go for the 3 days or for the 7 days. And this has been adopted from the Philippine Society of Microbiology. Because this leptospirosis, as I said, it's very common in the marsh area, like in Vietnam and Philippines, in Thailand, in Taiwan and Mongolia, in other areas, the adjacent area of the Hong Kong and the China, in Pakistan, where there's a huge area, like in the upper part of the Kipika and all that. That was, I think, all about the Leptospira. Thank you very much for your time, and we will meet again and we'll see what we have to do at the next time. Thank you very much.